All right. Good afternoon to everybody. Thank you so much for joining us today. We'll give it just 1 or 2 seconds for it to sync here. I'd like to welcome everybody to our roundtable discussion this afternoon. We're very excited to have our panel with us. The topic today is advancements in genome integrity assessment for vector-based gene therapies, and I think we're going to have a really great conversation. Just a quick first order of business, a legal disclaimer. The things we're talking about today are intended for molecular biology use and not for diagnosis, prevention, or treatment of any diseases. Just to give a little bit of background about our topic today, gene therapy has been around for quite a while now, from the initial proposal that the idea that you can replace defective DNA with a good copy to try to treat or cure disease. It's slowly built up into practicality. In the last 5 to 10 years, have seen a huge acceleration in advancement and development, with culminating in several FDA-approved gene therapies now. And this acceleration has brought about the need for an improvement and advancements in some of the analytics that are used in the AAV production process. There's some upstream with the plasmid development, the transfection and actual production of the cells, and then more the downstream where we talk about extraction, processing. And what we want to focus on today is some of the analytics and genome quantification. The AAV genome itself is what mediates the transfer of the transgene and basically gives the functionality for the gene therapy. So it's an important piece of that. And so that's what can be used for detection. So having it properly quantified is extremely important for safety and efficacy, to make sure that the dosing is correct. Because it's DNA, we can use PCR to do that quantification. In theory, it seems very simple, but in practice, there's actually a fairly complex AAV genome population involved in this. As you can see here, we have the vector genomes. Sometimes you'll have a full genome, and sometimes you'll have a partial genome. And you also have some DNA impurities in there that you need to sort out. So there's a great need for an improved analytics and characterizations to be able to determine that we're actually having a functional and a proper gene therapy product, an AAV, that is what you think it is. So to help us with this discussion today, and especially with this integrity piece, to make sure that we have to compare the full versus partial, we've assembled a panel of experts. So from Agathos, we have Andrei Noishevsky and Alex DeLilio, scientists there. From NEBA Labs, we have the Chief Scientific Officer, David Dobnik. And from SK Pharmteco, we have Robert Hills, who is the Director of Molecular Cell and Gene Therapies. And so with that, I would like to thank our panel for joining us today. And we'll go ahead and throw out the first question here. So Rob, you're involved with production of AAV. Can you tell us why the vector integrity and accurate quantification of different fragments of the vector genome are such important parameters to assess?
Yeah, that's a really good question. So I think the main reason is efficacy of the drug product itself. So, we are constantly dealing with a battle of getting, there's the whole issue of full versus partials. And then, then now trying to understand what those partial sequences are. And do they play a role in efficacy? Are they fragments? Are they truncation, or essentially truncations or splice variants and things like that? But I really think it comes down to knowing exactly what we're dealing with in fully characterizing our drug products. We can take that information back to R&D to begin working on maybe sequence optimization, or maybe there's really nothing we can do in the production side of things as far as guaranteeing full, essentially controlling partial sequences. But there is a lot that's kind of coming out in the literature now of identifying, like, essentially promoter regions and such that are prone to essentially splice variants or nicking or non-homologous end joining. So we can use that information to help us design better AAV constructs.
Great. So, David, NEBA Labs kind of focuses in a lot on some of these analytics. What are some of the available solutions today to do these analytics, and what advantages can digital PCR bring to that?
Well, in terms of determining the vector genome integrity, there are several options, and some of them are fairly simple. It can be simple gel agarose gels, so it can be capillary electrophoresis. However, those two, let's say, are not really quantitative, and you see some kind of nucleic acid, but you don't know what is inside. Then, of course, if you go to really more specific methods, long-read sequencing is fairly popular for this purpose. However, it has a bit lower throughput. Let's say, it can be quantitative more in a relative way. You cannot determine actual copy number of the fragments that you have. However, in terms of resolution, you get the complete sequence of the fragment. And this can be quite important. But then, of course, there's digital PCR, and digital PCR offers absolute quantification of complete genomes and also fragments. It can be really high throughput method. And at the same time, you can get results much faster. And just to show you what's already available, let's say on the market, we have a 4-plex assay that can be used for quantifying vector genome integrity. And you can get information on the completeness of the genome and, of course, the absolute copy numbers. So digital PCR, in a way, I think will kind of shift this field of vector genome integrity in the direction of absolute quantification. And with that, we could make quite a lot of interesting outcomes in the end in terms of correlations with other results like potency.
I would add here, if I can. One thing you mentioned about the sequencing, and yes, it's a very good detailed description of the genomic content of the AAV. However, I would say no matter what you do, I mean, it would take time consuming and way more expensive. And here I see digital PCR as a tool which can actually kind of help us to, assess the genomic integrity, maybe even with a slight less detail. However, the time of, assessing this, while you're producing AAV, doing a simple PCR reaction or dPCR reaction versus, full-blown sequencing. And that's actually a great advantage, I would say, here for an assessment of integrity using the dPCR versus sequencing. And obviously those two methods, I would say, can help each other. But yeah, that's how I see it, actually.
The benefits we've also seen from the digital PCR is the high sensitivity. And so with some of the methods that David mentioned, like agarose gels, you just don't have the sensitivity to see a discrete species of the AAV genomes in the population. So we have benefited from that with the digital PCR as well.
Yeah, and I have to agree with pretty much everybody's assessment. We do offer essentially long read sequencing, but as it's pointed out, it is a time-consuming effort. Digital PCR is definitely a very fast way to get to the endpoint of looking at genome integrity, compared to other methods such as agarose gel electrophoresis or, potentially, trying to correlate AUC values or something like that to your efficacy.
Question to Rob real quick.
Mm-hmm.
So I was just curious, we have just gotten started on the sequencing side of things. Have you seen any type of systematic errors in long read or short read sequencing that still need to be ironed out before that becomes more of a pipeline?
Well, I think with the short read, you're not going to get really, you're going to get a picture of everything that's there. So you're not going to be able to actually piece together you can piece together a full genome, but you're not going to know if they're actually contiguous sequence and it's a full length. I would have to go back and to answer your question. I mean, we've just gotten into doing long read AAV sequencing. So this is still a current method that is in development here at SK.
That's great, thanks. So David, just going back, so we see we have these multiplex assays up here. What are some of the considerations when you're trying to design a multiplex? What are some of the challenges of trying to put something like that together?
Well, first one is definitely the selection of targets. If you want to evaluate integrity, you need to have, let's say, assay that covers the whole vector genome. But of course, then it's on you to decide whether you're interested in the specific, let's say, regions like enhancer, promoter, poly-A, or you just want to have, let's say, every 1,000 base pairs another assay. But essentially, you need at least the beginning and the end covered if you would like to have this completeness of the genome evaluated. Then, when you have the target, as let's say our experience go, it is important which oligos are designed and which are selected, because we have seen that oligos, let's say differentiating with each other just in a few base pairs, can have really different results in terms of efficiency of amplification. And with that actually comes the second step, that is interaction between the oligos. And those can be quite extensive, and somehow in silico predictions are not always possible. So in silico evaluation might give you kind of really nice score, but then when you try everything in real life, put everything together, then you get quite some interaction. So definitely the design as such is a very important part of the assay. And then in the end, on the other hand, is also, let's say, after optimizations, data analysis that is the challenge in a way as well. So since we've been doing that now for 3 years already, at that time, this kind of integrity application was not even intended with the software of the producers of digital PCR platforms. And we just had to come up with some ideas of how to analyze this kind of data that we get. And at least QIAGEN was at that time definitely leading, at least with the multiple occupancy readout that the software provided.
That's what I wanted to say, was that they just, the recent support for multiplexing and for multiple occupancy and the intactness percentage with the intactness percentage error, it really supports this type of work, not just for AAV integrity, but also just for any type of integrity measurement in the QIAcuity.
So on that line, what would you say is the hardest part of doing that, of the analysis? What's the biggest challenge?
Well, of setting up or of the data analysis?
All of it.
Because the setting up, now that there's the crosstalk matrix, in this, it does make it quite simple. I mean, one of the issues, like David was saying, was once you get all your parts and your pieces, sometimes you'll have issues even if in silico it made sense. And a big one there is the bleed through. And so being able to cut out that bleed through makes it a much cleaner assay. So the data is a lot cleaner, and your thresholds are drawn a lot better. So at the end, it does make for an easier data analysis.
Yeah, I must admit here that this newer software version with this color compensation matrix definitely helps, because we have seen in the past there was some bleed through with channels, and then suddenly instead of 4 channels in 2D view, you're getting 6, and they're positioned in various ways. And then it became quite complicated for, let's say, unskilled user on where to set the thresholds appropriately.
Yeah, especially if we're going to start making, 8-plex multiplex assays with the new higher order multiplexing, supermix. At that point, it's nice to have that mixture as well, just because we were starting to see some, assay competition getting past, 3 or 4-plex even.
Ah, I see. So, Rob, this kind of then rolls into you from a user perspective. What are your expectations for this kind of assay? What would you need to see a multiplex assay for integrity provide?
Okay, so that's a really good question. So, I mean, I run essentially the GMP operations for molecular testing here at SK. And I think one of the biggest things that I look at when I'm essentially looking at assays that we may want to begin implementation of is essentially simplicity. That is like one of my number one priorities, because as you know, if you have too complex of an assay or too many steps involved, your precision can start to get hammered a little bit, because each step is going to represent some variability in it. So simplicity is number one. I look for robustness too. I mean, essentially we would put such an assay, if we bring it online, we would qualify it. So we would look at essentially precision, accuracy, linearity, specificity, those parameters to actually assess and really put the assay through its paces before we can actually stamp it that it's GMP ready. But I mean, those are my two biggest concerns when bringing on a new assay in-house. We have done, I think I will agree with everybody, with the new data package or analysis package that's been released by QIAGEN that has made our own studies have been conducted by Hong Park in my lab. We were originally calculating integrity by hand. Now we can do it with the instrument. So I look for things like that, just really simplicity and robustness of the assay.
Right. So shifting gears just a little bit, what are some of the, and I'll ask this to Rob, what factors can affect genome integrity? What do you have to keep an eye on in production to make sure that you're getting an intact product?
So speaking with my R&D colleagues here, there's really not much that you can do when it hits production at that point. I mean, you could take a radical approach and lower the temperature, maybe get better fidelity of replication, but you'll take a massive hit on your overall vector titers at the end. I think one of the big things is, to consider, and with the wealth and essentially people starting to generate long-read sequence data, the inclusion of new AI tools for modeling that data, a lot comes down to actually the sequence itself as being one of the major drivers that leads to this genome stability or integrity issues that we're seeing. I mean, a lot of it happens in the promoter region. Promoter regions are very GC rich. You can get short hairpin formation, which can lead to replication stalling. There's nicking that's been identified that can lead to non-homologous end-joining of sequence.
Oh, I think we might have lost Rob there, I'm afraid. Does anybody else have anything that they have seen in their work with AAV? I know Agathos, you guys.
Perhaps I can add.
I mean, it's a great question.
Oh, sure.
Sorry.
From the results that we have seen with the genome integrity assays, definitely what Rob mentioned, like packaging and sequence, is probably the kind of the thing there. Because let's say we have even seen that in some cases, some samples, packaging from one side of the vector genome was more effective. So we got, let's say, complete genomes, but then from the other side, there were like fragments kind of coming closer to the end of the genome. So let's say 5' packaging, 3' packaging can be different. And this is kind of done randomly in a way. So those factors definitely can affect what you get.
So from my perspective, I would say from perspective of the basic science, I would say, genomic integrity, I would say, could be impacted by multiple parameters during the production. And let's say being able to assess genomic integrity relatively quickly and cheaply, while I'm changing different parameters, either upstream or downstream, would allow me to sort of optimize it for the best results if those are impacted. So in a sense, like I said, that genomic integrity with dPCR somehow become easy and cost-effective tool to observe those changes instead of always going for the, let's say sequencing. And how to say, when we find some differences, then we could assess them with the sequencing, in a sense, what actually happening there. So that's kind of from a perspective of basic science production. So.
And Rob, welcome back. If you could please finish the thought that you were on.
Where was I cut off?
You were, well, we'll just go back from saying that the R&D said that there wasn't, you just start back from there. R&D said there wasn't a good way to tell.
I mean, no, so at least with production of the AAV itself, there's really not a lot of factors that we can essentially work through to aid genomic integrity and full integrity. I think a lot of it all comes back down to the actual sequence itself. There's been a few papers published out there showing that you can get replication stalling at promoters. Promoters tend to be highly GC rich. So you run the risk of possibly forming short hairpins, which can lead to replication stalling. Or there's also replication forks. There's nicking has been identified in the literature that leads to non-homologous end joining. So I think as more and more long read sequence data is produced and using some of these newer AI models, I think it really comes down to optimization of the sequence. One of the other things we've seen at the bench level in performing our own integrity analysis is the actual handling of the AAV drug product itself. We've seen that the capsid lysis step, there are critical parameters around that as far as either using temperature or protease K. We have seen that they also lead to differences in the integrity values that we get. So, I mean, we're diligently trying to optimize those steps. But again, I think it all really comes back down to the sequence.
Right. Thank you. So this is a question for Andre. So I know Agathos has developed kind of a universal AAV standard. How is it designed to be able to enhance its utility in AAV research and production? How can it help with integrity analysis testing?
So one of the reasons actually behind it, when Rob mentioned about simplicity and robustness, of how to say assessing genomic integrity from his GMP perspective, when I also was looking for a basically way to handle properly, assay development as far as AAV quantification. And when I was started working with AAV, I looked into various positive controls as far as the AAV out there. And yes, they are there. However, the limitation was that number of targets commonly used in cell and gene therapy was not there. In a sense, commonly used is ITR, and a few of those targets, promoter kind of, and then poly-A, which could be found in various gene of interest. But when I started working with more types of gene of interest, what started happening that I couldn't find, good representation of the assays I was using. And occurred to me that it'd be good to have a tool which kind of is relatively flexible as far as what assays I can test on that gene of interest. And here the QIAGEN came with the cell and gene therapy assays for dPCR, with basically designing designing those assays for those targets. And I decided to place them on the as a gene of interest, basically, in an AAV. And that way, basically, I can start using those and comparing the titers across those targets and see how those assays perform. And knowing that quantification of AAV is highly dependent on the, like Rob mentioned also, for example, what kind of lysis are you performing? What's your sort of protocol for preparation of AAV for a quantification? And now having multiple assays on the same template, I can see how well they perform and also start comparing those results with other products which are floating in our laboratory. So that actually, I would say that simplicity and universality of that template kind of helped. So and I would say, and in time, this tool became relatively valuable in a sense. We could actually start assessing genomic integrity because now we have a multiple well-characterized assays developed for those targets across the template of the gene of interest as far as the DNA. And we don't have to necessarily use the longest fragments, but we can start looking at the shorter and various different types of populations of those DNA, which is encapsulated in AAV. So kind of give you the more detailed description of that content. And so that's kind of like a basic idea in a sense why that standard exists. So it's not just necessarily a positive control for a titer using those multiple targets, but also it can be used as a positive control for genomic integrity studies. So let's say you are analyzing your gene of interest, whatever that is, but then, like I said, the process of titering of AAV is relatively complex, and then you have to analyze the data as far as your genomic integrity. So now you can actually use this control as a positive control for genomic integrity and check your math, let's say, too, if all the time your assay agrees. And so that's one of those aspects, I would say. And again, the standard have a extra space, I would say. DNA staffers were added there, so we can keep the same size for the future use, but we can start adding other commonly used fragments to basically expand that universality of it. So. And as you can see here, we are relatively happy with how those targets amplify across the genome. So that's, I would say, it's a sort of basic rationale between this, to find a tool that is easy to use and relatively flexible. And yeah.
And can you go a little more detail about how those multiple sites and how that's useful for assessing integrity?
So let's say, actually, I will go to the next slide here. So one of the preliminary results, for example, in this case, we just use a duplex, and there are 4 different duplexes we designed assays. We ran it, and as you can see, first duplex have fragments which are relatively close to each other. Then second duplex, the fragments are, the targets are a little bit farther from each other, and so on, to almost the last one to spanning almost the whole coding sequence of the gene of interest. And using those 4 sets, we were able to assess a proportion of those fragments in the AAV population. So here you can see that the largest fragments, about 3,000 nucleotides, were about 48% of the whole population. So that will tell you that, let's say in this case, the intactness, the useful, coding sequence of in this case, EGFP, was at 48%. And then other fragments with the shorter lengths, different percentages of representation. And like I mentioned before, yes, we could perform sequencing, and that was done already to basically cross-validate those results as far as genomic integrity versus sequencing. But in this case, we have that representation of those different lengths of fragments without actually doing it, and data was obtained relatively quickly. So that's what I would say. And again, standard could be used in this case, as a positive control for those assessment of genomic integrity, accompanying the assessment of the actual therapeutic product integrity.
Right. And can these be used with qPCR as well? Is this limited to just digital?
I would say...
I think I might be able to jump in on that. that one, yeah. Yeah, so we definitely can use it for qPCR in-house. We really try to utilize it for any method, especially newer methods. And as multiple people have said previously in this conversation, it's important to have good positive controls, especially as you're developing new methods. So not just qPCR, but many other methods we try to use it for. One of the best things when using it for qPCR is that we do have a catalog to pick from. So if we're going to do a tech transfer or something like that, it's easy to just point our clients to the QIAGEN website for them to be able to buy the assay. And then having the universal AAV standard means that we can get it in their hands to assist them in the tech transfer. Additionally, in-house, like I mentioned, we do automation for our titering as well as genomic integrity. And it was a very valuable tool to have for that, as automation is a little bit of a pinch point if you're going to be doing genomic integrity, just because, yeah, it is a little rougher than human hands.
Yeah, but I would say that the dPCR comes into the place where you can actually start looking at those multiple targets amplifying in a one nanoparticle partition, basically, where actually you can start actually taking out that data from to assess the genomic integrity. So I don't think so. qPCR is at this point capable to do it. As far as
and we really do try to leverage it for digital PCR because at the moment our in-house assay gives us genomic integrity information at the same time as titer. So all of the samples that we test, we're getting genomic integrity information on, and it's almost fully automated, which is wonderful.
That actually, oh, go ahead. Oh, okay. So that actually kind of takes us into our next question. I'll throw this one out to Rob. So what are some of the orthogonal methods that you would use to verify these results from digital PCR? I know you mentioned some sequencing and just mentioned qPCR. What's the correlation of that between those results and what you've seen with digital?
So we don't have a huge data set right now. I mean, we've just gotten into this. So, I mean, I could defer to David on this question. I mean, we do have essentially anecdotal evidence of what we're providing as far as integrity matching with what they're seeing possibly in cell-based potency assays. But we haven't done a side-by-side comparison of essentially taking the AAVs that we've worked with for integrity mapping by dPCR and comparing that to essentially long read data that we've generated on those same sequences in-house. So maybe David would be better suited to answer this question.
Yeah, well, I can add some additional information, of course. I must tell you that when we first run these integrity assays on AAVs, we were very surprised because the values that we were getting in terms of numbers of complete genomes were very low. So they were like from 20 to 40% of, let's say, whole populations of different genome fragments. And this was like difficult to believe in a way, especially if you're seeing that the samples are getting like 95% full from AUC or other, let's say, full empty evaluations. So it was difficult then to say whether the assay that we have is really good or not. And since it worked really perfectly in this, let's say, synthetic environment with double-stranded DNA, whole-length genome, we fragmented it and it worked well. But then when you see the AAV data, it was like very strange. And of course, it was very difficult to get some kind of orthogonal methods for us at that time to kind of see if there's any correlation. However, we had then a really nice collaboration with a team from AskBio, and we designed assay for them, and they did check those values against the NGS data, and actually they got really nice correlations with the integrity. So I think these kind of correlations are the most essential because both are focused on the genome, whatever we are looking in terms of, let's say, correlating it to the full and empty, those are not as good anymore. And yeah, I think correlating the data on the integrity then to the potency, this is kind of the most important thing. And there was a recent publication that has shown that when they were titering the AAVs and just doing potency based on the titer, they got different results in potency. But when they checked the sample in terms of the integrity, they've seen that the samples are actually different, and they could really nicely correlate the amount of complete genomes to the potency result. So definitely there is kind of the assays, the multiplex assays for genome integrity in digital PCR can provide very relevant data. And you might even get an idea of what your potency would be in the end from such sample.
So, Rob, so you're not doing that. Have you seen anything like that? Any correlation between the integrity and the efficacy?
Well, when Hong and my group presented this data within the company, it was kind of pointed out at the end that essentially the R&D group that was running a potency on this same site, the same construct, actually saw a very similar effect in potency. Based on what we were reporting, I think we were reporting around maybe a 40% full integration, or essentially fully integrated genome, that they kind of anecdotally said this explains a lot of the potency data around this construct. So, I mean, this is something that we are trying to delve deeper into. We will be releasing a white paper next year where we look at different AAV constructs and then try to correlate that back to potency or with other methods.
Great. So one last thing I want to touch on here. So it's, we're talking about integrity, which is basically, whether something's being properly packaged or how much is being packaged. But kind of related to that is really kind of anything is being packaged or not. I know one of the metrics you're going to look at is going to be a full to empty capsid ratio. So, Rob, how do you guys check for that?
I mean, we use the gold standard analytical ultracentrifugation, but we've also got online and it's GMP ready mass photometry that we've been now using on a number of programs here at SK. And we see very good correlation between AUC and mass photometry. I know that there's workings out there about potential PCR applications for full versus empty. And I recently saw a very interesting paper on using essentially mass spec to determine full empty. Interesting thing is, if you're looking at the mass spec profiles, they look very much like a mass photometry profile. They give you very broad peaks, and you can see a lot of partials in there as well. I was actually hoping that mass spec had gotten to the point where maybe through high res mass spec, you can actually identify exact molecular weights of those partials, but I don't think it's there yet. But right now, like I said, we use AUC and mass photometry.
And Alex, what do you guys do at Agathos?
Yeah, so we do multiple different methods. One that we do would just be the regular digital PCR and ELISA, because we have that data to work with regardless. But we have recently started getting into using UV spec, SLS, DLS to look at the full and empty full, as well as other metrics that all give you, like aggregation.
And here is one aspect as far as for us, like Alex mentioned, ELISA dPCR, but being able to actually assess genomic integrity at the same time when we assess the titer, we actually can report basically the percentage of the full versus just the titer, yes, at the same time. So that's a kind of a relatively simple and cost-effective method to evaluate that. So
very interesting. Okay. Well, I think that's all the questions that I had. So I would like to go ahead and open up the floor to anybody in attendance. There should be a Q&A box that you can see. we have people working to look at these. We've had a couple questions come in, so I will ask the first one here. Yeah, touching on something that Rob had mentioned. So what AAV capsid lysis method is recommended to calculate genome integrity values accurately? Heat lysis or proteinase K?
Well, I would say right now, probably definitely proteinase K. We saw with heat lysis, we were getting fragmentation of the genome. So I would definitely go with proteinase K. I know there's thermolabile proteinase K. That probably would be a good choice because I know one of the steps that we use is we do a heat inactivation of the proteinase K. So we have been looking into the thermolabile proteinase K to try to avoid heating of the sample.
If I may add,
sure.
I would even recommend doing it without prior denaturation, because in that way you actually evaluate what was in the capsid, and once it is partitioned, you can get the signal from the content from the capsid. And yeah, as far as our experience go with prior denaturation, we have seen that yeah, we got, let's say, lower integrity of those samples just because you are introducing some fragmentation with the process of pipetting or perhaps even partitioning. So.
All right. Another question here. It is, are there other DNA molecules in the drug product that would benefit from an integrity analysis? Residual plasmids, for instance, or antibiotic resistance genes?
Well, we have actually addressed this already with the kanamycin integrity assay that we developed. And definitely, at least, let's say from my perspective, it's very important to know if you have the complete kanamycin resistant genes in your AAVs. If you just see fragments, okay, it's kind of impurity, but it could be much worse if you have really the full length gene inside that is going into the patient.
Yeah, at Agathos, we've used it to also look at the integrity of host cell DNA during DNA treatment, as well as DNA selection. So when we were shopping for DNases, we used that to confirm that we were getting below 200 base pairs of intact host cell DNA.
That's something that we look at as well, specifically around host cell is base pair length. I know there's a lot of requests now and potential regulatory ramifications about requiring a base pair length on host cell. So this is something that we've also addressed through multiplexing.
Right. Another question here. Would you use any normalization or housekeeping genes to normalize this data? Do you need some external standard basically to do these calculations?
For the integrity as such, I would say no, but perhaps the question refers to, let's say, other applications, like if you're checking about, let's say, integration or when it is in the cells, so kind of biodistribution, you might want to check whether you have intact genome kind of there in the cell, but together you want to see whether how much cells you have, and then let's say what's the amount of genomes per cell. Then you can use some kind of normalization to kind of just get the number of the cells at the same time. But for the genome integrity, it's just absolute quantification. You don't need any normalizator.
All right.
Yeah, the only place I chime in there is if you want to include an internal control. If you have enough room in your multiplex, would be beneficial sometimes if you can fit it in.
All right. So let's see. So is there a specific DNase that you recommend to digest the host cell DNA that you just mentioned?
So we rely on the vector lysis kit, the QIAGEN the vector lysis kit, viral vector lysis kit that launched about a year or two ago, and we get good results with the DNase that's included in that kit.
Anybody else, anything specific they like?
We have in the past on one program used turbo DNase with good, essentially with good results.
I don't see any other questions here. We'll give it a minute, see if somebody has something. Does anybody on the panel have any kind of last statements or anything they'd like to
I'd like
address?
to clarify the DNase point, if that's okay. So
Sure.
When I mentioned that, I wasn't talking about the DNase during the dPCR sample prep. I meant when we were shopping for our DNase for production. So that wouldn't be our polishing DNase for the sample prep. That's our bulk DNase for harvest, just to clarify.
I see. All right. Well, if there's no other questions and nobody has any other statements, then I'd like to go ahead and close it out here. I really appreciate everybody's time today. It's been a great discussion. I've learned a lot. I think it's been very valuable. If there are any questions that come in late, just please feel free. There's contact information for each of our panelists here. Just feel free to reach out. Well, I do see one more came in. Okay. So what part of the dPCR dynamic range can be used for integrity analysis?
Well, essentially, this depends on the platform. So definitely with QIAGEN, I think you can use highest dynamic range because they have the specific analysis that enables quantification and also takes into account the colocalization of several targets, not just, let's say, in duplex format. But otherwise, yeah, you need to take care on the colocalization. So the dynamic range, let's say, might be lower than for the normal simplex analysis.
All right. There's another question here. So would a capsid viral delivery affect the heart organ adversely? That might be more of a clinical type question, but we can maybe put that to refer. If nobody wants to jump in. We can follow up with that one. Don't see any others. So with that, I guess I would like to go ahead and close out. Again, I appreciate everybody's time and for everybody attending. If you have any questions, feel free to reach out and check out those controls. You can get those controls from Agathos, and Niba Labs has their entire set of assays for this, and of course SK Pharmteco, full service there. And with that, I'd appreciate everybody's time, and we will see you at our next webinar.
Great. Thank you. Thank you.
Thank you.